If you want a treat, you ask for one. Conversation is like neural signaling. It's a quick way to get a message across to one or two people. But what if a more far-reaching signal is needed? When an airplane is about to encounter turbulence, instead of telling each passenger individually, the pilot broadcasts an announcement throughout the plane. Hormones are a lot like a pilot's announcements. They broadcast information throughout the body. These chemical signals diffuse from one cell to another over long distances to potentially reach all the cells. Just as all airline passengers may not respond the same way to an announcement from the pilot, all the body cells won't respond the same way to the message announced by a hormone. Hormonal signals are usually delivered by the circulatory system, where they are carried to all parts of the body. Although the signal may be far-reaching, only certain cells may receive and act on it. Cells that respond to a hormonal signal have specific receptors for that particular hormone. These cells are called target cells. One hormone can cause different responses in different types of target cells. For example, the same hormone may cause one type of cell to contract, another to secrete, and another to divide. The study of hormones is called endocrinology. Strictly speaking, an endocrine hormone is a chemical signal that's secreted into the bloodstream from a gland without ducts, an endocrine gland. But as we'll see, not all hormones are secreted from glands, and some don't ever reach the bloodstream. Instead, they might travel short distances in the fluid between cells. To survive, organisms must be able to sense changes in their external and internal environments and respond accordingly. The nervous system coordinates activities like movement, which require specific and rapid responses that take place within seconds. Hormones integrate more inclusive and slower responses that may take place within minutes, hours, or days. Hormones coordinate the activities of all the body's cells, from the maintenance of homeostasis to the timing of the events of reproduction and development. Important cues from the environment, such as day length, temperature, and danger signals, can be translated into hormonal messages that help organisms respond. A special class of hormones, the pheromones, send signals between individuals of the same species. Sexual attractants are the best studied pheromones, but there are many others. Silk moths secrete a pheromone called bombicol. Male silk moths will follow the path of bombicol up its concentration gradient for several miles to find a female. The man shown here has a queen bee under his chin. She is releasing pheromones to promote swarming. His bee beard is the result. In this activity, we'll explore the chemical characteristics of hormones and look at the mechanisms of hormone action. Then we'll investigate some examples of how hormones act as chemical signals to integrate functions in an organism. Finally, we'll conduct an experiment to observe how hormones interact during insect metamorphosis. Although many different hormones have been identified, hormones can be classified into two groups. Hydrophilic, or water-loving molecules, include amines, peptides, and proteins. Lipophilic, or lipid-loving molecules, include steroids and prostaglandins. Hydrophilic hormones are soluble in water and mediate a faster response than lipophilic hormones. The receptors for these hormones are found on the surface of their target cells. The hydrophilic hormone insulin was the first protein whose amino acid sequence was determined. We'll learn more about the role of insulin later. Local regulators are chemical signals that act on cells very close to the site of release. They never reach high concentrations in the bloodstream. Nerve growth factor is an example of a peptide local regulator. This hormone is critical for the differentiation and survival of nerve cells. Lipophilic hormones, such as the steroid hormones cortisol and testosterone, consist of carbon chains and rings that are soluble in lipids. Their lipophilic nature makes them almost insoluble in blood, so they are bound to carrier proteins in the bloodstream. Lipophilic hormones pass through the lipid bilayer of the cell membrane. Once inside the cell, they bind to receptors in the cytoplasm or the nucleus. Thyroid hormones, like thyroxine, are lipophilic hormones derived from amino acids. They consist of 2 amino acids that are modified with iodine. This modification makes them lipophilic. Prostaglandins are lipophilic local regulators. They act on cells close to the cell that synthesize them and don't reach high concentrations in the blood. Retinoids also act as local regulators. You may be familiar with the use of retinoic acid to promote the local growth of new skin cells. Now that we can identify the different types of hormones, let's explore how hormones affect cellular activities. The hormonal regulation of cellular functions depends primarily on the specificity of the hormone receptors. All cells can potentially come in contact with a hormone, but only those that have a specific receptor for that hormone will respond. Although different types of cells may have specific receptors for the same hormone, they will respond to that hormone in different ways. In other words, the receptor decodes the hormone message, but the specificity of the response is determined by the nature of the cell that responds, not by the hormone. Hydrophilic hormones bind to receptors that span the plasma membrane. Different types of receptors transmit the hormonal signal to the interior of the cell in different ways. Some receptors, when bound by hormone, have an enzymatic activity that phosphorylates proteins in the cytoplasm to activate or deactivate them. The insulin receptor functions in this way. Other receptors activate or inactivate a separate membrane-bound enzyme or ion channel. This activation is mediated by a protein in the plasma membrane. This protein, which binds GTP, acts as a transducer of the hormone signal. Receptors that act via GTP binding proteins are called G-protein coupled receptors. Let's examine the most common action of G-protein coupled receptors, the conversion of the hormone signal into a cascade of second messengers inside the cell. When a hormone binds a G-protein coupled receptor, a cascade of chemical events within the target cell is initiated that usually increases the concentration of second messengers. A second messenger is a small molecule that relays the extracellular first message of the hormone into the cell. Proteins in the cell are then activated or deactivated by these second messengers. Because second messengers usually affect the function of proteins that already exist in the cell, responses are fast. Many different second messenger chemicals have been identified. The most common are calcium ions and cyclic AMP or CAMP. Second messengers both convey and amplify hormone signals inside cells. One hormone-binding event on the cell surface will activate many G proteins. In turn, these G proteins will send subunits to activate many adenylate cyclase proteins that convert ATP to CAMP. A hormone-binding event is then greatly amplified by the production of many CAMP molecules. The system is designed to automatically shut itself off. The activated adenylate cyclase is shut off when the G protein subunit dephosphorylates the bound GTP to GDP. The subunits reassemble, ready for activation by another hormone receptor binding signal. The hormone receptor signal is terminated by endocytosis of the hormone receptor complex. The receptors are then recycled back to the cell's surface. Often the number of recycled receptors decreases following exposure to the hormone. This decrease in the number of cell surface receptors is a form of downregulation. Now that we have looked in detail at one of the most common mechanisms of hydrophilic hormone action, let's turn our attention to the mechanism of action of lipophilic hormones. Lipophilic hormones pass through the cell's plasma membrane. Once inside their target cells, they encounter specific intracellular receptors that share the following structures: an activating domain that binds to other transcription factors, a DNA binding domain, and a hormone binding domain. When bound by hormone, the hormone receptor complex changes shape, enabling the protein to bind to regulatory regions on specific genes. The bound hormone receptor complex attracts transcriptional regulators, which then activate or suppress the transcription of the genes. In essence, the lipophilic hormone receptors are hormone-activated transcription factors. The products of some genes may change the function of the cell. Others may, in turn, activate other genes for a delayed secondary effect. Because it takes time for proteins to be synthesized, lipophilic hormones are best suited to controlling long-term processes such as the events of reproduction. As long as the hormone concentration remains high, its receptors will probably have hormone bound to them. When the hormone concentration decreases, as hormone molecules dissociate from the receptors, it's not likely that another hormone molecule will be available to take their place. The activation then ends. Now that we've learned about how hormone action is initiated and terminated, let's look at some specific examples and explore ways of regulating hormone action to affect blood glucose levels. Endocrine cells receive information about the external and internal environments. We'll examine some examples where the need for additional blood glucose is regulated by different hormones and by different mechanisms. Sometimes an endocrine cell is also the sensing cell. In other cases, the information is relayed to the endocrine cell in a variety of ways. In the body, nerve signals directly stimulate the secretion of the hormone epinephrine in the fight or flight response. For example, a deer's sensory systems send a nerve signal to the brain announcing danger. The brain triggers sympathetic neurons to stimulate the adrenal medulla, which releases epinephrine. Epinephrine binds to receptors on the surface of many cell types to prepare the body for quick action. By way of second messengers, epinephrine activates enzymes in the liver to break down glycogen and fat cells to release fatty acids for energy. The heart beats faster and stronger, and the smooth muscle of blood vessels that supply the intestines contract. Blood glucose levels rise, and the delivery of blood glucose to the muscles and brain is maximized. Some nerve cells secrete hormones directly. When stimulated, certain neurons release hormones called neurohormones. Many of these neurons are found in the hypothalamus, and their axons usually terminate in the pituitary region. Here's an example. During finals week, you may experience prolonged stress and anxiety. This longer-term stress is conveyed to neurons in the hypothalamus of your brain. These neurons release corticotropin-releasing hormone, or CRH, into the bloodstream, which serves the anterior pituitary. There it binds cells that have its specific receptor, causing the release of adrenocorticotropic hormone, or ACTH. ACTH is a tropic hormone that causes the synthesis and release of another hormone, cortisol. ACTH travels via the blood throughout the body to the adrenal cortex, where it binds to cell surface receptors and initiates the synthesis and release of the steroid hormone cortisol. Because cortisol is lipophilic, it can enter every cell as it travels in the bloodstream. In target cells, like liver cells, cytoplasmic glucocorticoid receptors are bound to proteins. When cortisol binds the receptor, these proteins release, allowing the hormone receptor complex to move into the target cell's nucleus. In the nucleus, like most lipophilic hormones, cortisol receptors will bind as dimers to DNA. The hormone receptor complexes bind to specific regions of genes called response elements. Within minutes, the transcription machinery is assembled and gene products are made. Cortisol maintains proper levels of energy substrates partly by increasing the production of enzymes that synthesize glucose. This helps provide the body with adequate levels of blood glucose during times of prolonged stress. Our body needs an adequate supply of blood glucose for maintenance and occasional bursts of activity. However, too much blood glucose can be detrimental. Although the way our cells sense blood glucose levels is not completely understood, a low-affinity glucose transporter might be a glucose sensor in pancreatic beta cells. Glucose sensors in the hypothalamus, brainstem, and liver also play important roles. These sensors guide the interplay between two antagonistic hormones, insulin and glucagon, to maintain proper blood glucose levels. Insulin is secreted by pancreatic beta cells located in cell clusters called islets of Langerhans. Insulin receptors are found on almost every cell. Proteins in target cells are modified to prevent the synthesis of glucose and promote the synthesis of glycogen. Insulin also stimulates enzymes in fat cells to synthesize fats from fatty acids and glycerol and stimulates protein synthesis in liver and muscle cells. The action of insulin is terminated by endocytosis of the hormone receptor complexes, and the number of receptors can be downregulated upon continued insulin stimulation. Glucagon is secreted in response to low blood glucose levels. Made by the alpha cells of the pancreatic islets, glucagon acts on the same targets like the liver, but with the opposite effects of insulin. Glucagon binds to a G protein-coupled receptor. The resulting cascade of second messengers leads to modifications of proteins to break down stored glycogen to release glucose. It also stimulates glucose synthesis in the liver and kidneys. The release of glucagon is inhibited by insulin, which rises when glucose levels in the blood are again high. Insulin, glucagon, epinephrine, and cortisol can all affect the level of glucose in the blood by different mechanisms and in response to different stimuli. The absence of an insulin response results in the disease diabetes. Diabetes can be caused by a defect in the production of insulin, or defects in the receptor-mediated response to insulin. These graphs represent insulin, glucagon, and glucose levels in the blood of two people during a short fast. Click on each graph to see what happens when they drink a sugary soda. Can you tell which person is diabetic? Yes, good job. The diabetic's blood glucose level remains high for much longer than normal. If left untreated, gradual damage to organs such as the kidneys and eyes will result. We've seen how different hormones affect the amount of glucose in the blood. Hormones can work together to coordinate the response to the many demands made on our body. Hormones are secreted chemical messages. They can act on cells far away or very close to the cell that produced them. They can be proteins, single polypeptides, amines, or lipids such as steroids. Hormones usually travel to their place of action via the circulatory system. They bind to specific receptors inside or on the surface of their target cells. The same cell could be a target for many different hormones. Receptors for hydrophilic hormones are located on the cell surface. The binding of the hormone causes changes in cellular activity, directly or indirectly, by the production of second messengers. Enzymes may be activated or deactivated. Membrane transport may change, and gene expression may be affected. The end result is a change in cell function. Cell surface receptors bound to hormone are cleared by endocytosis. The number of receptors returned to the cell surface is often downregulated after exposure to hormone. Lipophilic hormones encounter their receptors inside their target cells, where they affect gene transcription. This change in the proteins within the cell alters the function of that cell. Hormones are released in response to changes in the internal or external environment. Sometimes the hormone-secreting cell is the sensor, as in the case of beta cells, and insulin. Other triggers for hormone release are neural, such as epinephrine, or hormonal, such as cortisol. hormone responses can be modulated by the action of other hormones, by feedback mechanisms that terminate the secretion of the hormone, and mechanisms like endocytosis that downregulate the hormone response. As insects grow, they develop a new exoskeleton under the old one. The old exoskeleton is shed in a process called molting. The insect fills the new soft exoskeleton with air and fluid to expand it before it hardens. The molting process is controlled by several hormones. Among these are the lipophilic hormones ecdysone and juvenile hormone. Ecdysone, a steroid, controls the growth of new exoskeleton. Juvenile hormone determines the appearance of the exoskeleton. Young cockroaches look like tiny versions of adult cockroaches, but they lack wings and can't reproduce. These juvenile animals, called nymphs, molt into larger nymphs until the fifth molt. At the fifth molt, they develop an adult exoskeleton with wings. Scientists have found that during each juvenile molt, the level of juvenile hormone is high. However, at the time the adult exoskeleton is being formed, the level of juvenile hormone is low. Ecdysone is synthesized in and released from the insect's prothoracic gland. Juvenile hormone is synthesized and released by paired glands, the corpora allata, often located just behind the brain. An insect can live for some time without its head. The location of the juvenile hormone-producing glands in the head of some insect species led to its discovery. We're going to reproduce some of these experiments by cutting off the heads of a new species of insect we've found. Our new insect species is a bug, and we've named it Rhodnius carpio. As we watch it develop from hatching to adult, we observe that it goes through 4 nymphal molts before it becomes an adult. We hypothesize that, like other insects, the levels of juvenile hormone in Rhodnius carpio are high during the juvenile molts. What do you predict will happen if we cut the insect's head off before the 3rd molt? Drag your prediction into the box, then click Submit. Without the corpora allata in the head region, there is no longer any juvenile hormone being secreted to maintain the insect's juvenile characteristics. We now apply synthetic juvenile hormone to a nymph before its last molt. What do you predict the outcome will be? Drag your prediction into the box, then click Submit. The addition of juvenile hormone will maintain the insect's juvenile characteristics. Instead of